My name's Ian Taylor, and I'm a director here at FlowJo. I can be reached at taylor@flowjo.com if you have any questions after this meeting. As I mentioned, the whole presentation will be posted to FlowJo University along with this slide deck if you want to download it there afterwards. So today we're going to be talking about differential expression analysis in SeatGeek, which leads nicely into the next lesson, which is gene set enrichment analysis. So that'll be coming up shortly as well in another presentation. But today I want to focus specifically on the differential expression of genes in single-cell RNA sequencing data sets, which you can derive from SeatGeek. What is differential expression analysis? Well, to put it very simply, it is a comparison between two populations with regard to the parameters, or also called features, that are distinguishing those two populations. We typically perform this comparison based on two statistical values, p-value and fold change. And that just leads us to the best possible results with regard to further downstream analysis. It also leads to a set of parameters which define a pair of populations' different features, and you tend to get both up and down regulated gene sets based on this analysis. Going forward, I'm going to be referring back to this example a lot, mostly with regard to the B cells identified here. Now, this data set was provided, I believe, by Stokius et al., who did some really cool antibody bar-coded sequencing work, wherein they tagged cells with antibodies bound to oligonucleotides as a way of barcoding those antibody-bound cells so that we could later identify the protein content as well as the whole transcriptome of those cells. So again, B cells here is identified by the canonical protein markers, CD19 negative, sorry, CD19 positive, CD3 negative. In order to begin differential expression analysis, we first got to define the clusters of interest, and this can be done in a variety of ways. One is by simply gating on the clusters, as we would see that in flow cytometry. Essentially, we draw a geometric gate in two-dimensional space that identifies a population of interest. Another possibility, especially in the single-cell RNA sequencing analysis world, is unbiased clustering. Now, unbiased clustering works by using some kind of algorithm to pick out populations that a human may not be able to or may not want to. We also often have categorical parameters which define things like disease state, sample type, etc., on which we can draw gates for categorical, what we call categorical parameters there. We can also utilize Boolean logic to do this, and I'll run through a couple of examples right now. So as mentioned previously for the B cell example, we can draw gates just like we would in flow cytometry analysis in FlowJo. In this case, I'm using an auto gate to define different areas of high density within a t-SNE mapping. So we've just created clusters, it's like 1 through 7 in a t-SNE mapping, or sorry, 1 through 6. And this is somewhat unbiased because we're simply drawing gates that align with the contour lines of the t-SNE mapping, but we still have some judgment calls to make, and so it's not completely unbiased. If we wanted something a little bit more unbiased, we would want to use an algorithm such as k-means. In this case, I'm showing the output from a Seurat pipeline run on a particular data set. And the Seurat pipeline is developed and maintained by the Satija Lab at NYU. It's a really popular and very powerful way to gain insights into a population of cells that have been single-cell RNA seqed. And we get quality control plots here on the left, showing violin plots, indicating the coverage of expression for each of the clusters identified. On the right, we see that the Seurat pipeline has developed t-SNE-X and t-SNE-Y parameters as well as these graph-based clusters. And again, that's the unbiased clustering that we're referring to in this case. We also have categorical parameters in this data set, which was provided by the AMP group for lupus patients. So we can see that they've included control B cells from unaffected kidneys, as well as those from patients affected by lupus. And so we're able to very easily gate on control data versus test data with the disease state in this case. And any of these populations, just to kind of foreshadow where we're going with this, any of these populations could be used for differential expression analysis downstream. But the first step is to isolate some clusters. Finally, a really popular or really useful tool for gene set or differential expression analysis is the Boolean NOT gate. So if we're interested, in this case, of course, in B cells, we would select B cells from within the workspace in the samples area, and we could just go to the Boolean ribbon and choose Make NOT gate. That will then create a population called B cells minus, indicating that it is a NOT gate for B cells, and I've renamed that NOT B cells to be clear that what we're dealing with. And so these are going to be all cells that were not identified within the B cells gate for use as a comparator in downstream differential expression analysis. So really handy to keep that option in mind. Of course, there's all sorts of other Boolean gates that can be utilized. Some of them are probably less useful for the differential expression analysis that we're focusing on today. So with that, let's get into another really important area of analysis within SeatGeek for doing differential expression analysis, which is the gene view. Now, for those of you who are, again, FlowJo users or more accustomed to flow cytometry data analysis, you will be very accustomed to cell view, wherein every dot within a dot plot is a single cell. In this case, if we click gene view within the graph window, we're actually going to visualize the parameters or the genes themselves within each of those populations. In that case, the parameters are no longer the genes themselves because, again, we're visualizing them graphically. Instead, we'll be using populations that we've identified as the parameters. And this is actually a really ingenious method, I think, of getting at information about the expression values for those given populations. So without further ado, let's look at a little bit of gene view. So again, cell view, we're isolating clusters, as we see on the left here. And we have a particular expression matrix, which is divided up into cells and genes. If we sort of transpose or tilt that matrix onto its side, we now have clusters being identified versus their genes, as opposed to genes per cluster. And in that case, we can place clusters 1 and 2 on the X and Y axes. And when we gate, we're actually gating on the parameters that define those clusters. This is something that we call a pivot analysis. And I'll get to a more functional way of getting differential expression in just a moment. But just to illustrate that in SeatGeek, here we have a couple of samples identified from within a 5-sample concatenation. And we can see that sample 5 has been placed on the y-axis, sample 4 is on the x-axis. We're viewing this in gene view, and so when we draw a gate, we get a gene set, which I'll talk a little bit more about in a moment. We get a gene set identified in the workspace here with 3 genes there, 1, 2, 3. So that kind of makes intuitive sense, I hope. Again, if you have questions, feel free to send them along to seatgeek@flowjo.com or to me, Taylor@flowjo.com, and I'll be happy to help you out. If this is a little bit confusing, I totally understand. So in order to begin differential expression analysis, we want to make an initial comparison. In that initial comparison, we're going to place a control population on the x-axis and a test population that we're interested in learning differential expression information about on the y-axis. Usually, once we've provided that initial comparison, we don't want to draw any gates. Instead, we want to gather the fold change in p-values that I've mentioned previously. Here's an example of that initial comparison, wherein we're showing not B cells. These are our control this is our control population, versus our test population, B cells, on the y-axis. Now, again, looking at that initial comparison, if we want to get differentially expressed genes, what we really want to do is open up a volcano plot. And a volcano plot is accessible in SeatGeek by clicking on this differential expression of genes button here, or the DEG button, and that will open up a volcano plot. In this case, we're not actually looking at B cell differential expression, but this is just kind of a very pretty example of the volcano plot in action. And what we immediately notice are arrows pointing up and down. The up arrow indicates these are upregulated genes in this given comparison, and the red arrow pointing down shows those genes that have been downregulated in this comparison. Along the x-axis, we have fold change. Anything that's positive for fold change is our upregulated genes, and anything negative for fold change is, of course, downregulated with regard to our comparison, or our comparators. We also have a y-axis that shows an adjusted p-value, also called a q-value, and that's performing a Mann-Whitney U-test, two-tailed, with a Bonferroni correction, in case you're curious. And of course, as we move up that scale, closer to 0, we get more and more significant comparison, more and more significant genes with regard to our comparison. And as we move closer to 1 at the bottom of that inverted log scale, we are getting less significant parameters with regard to our two comparators, i.e. Less reproducible. That's the prediction that's being made. Here's an actual example relevant to the example I keep coming back to for B cells, wherein I've gated those genes that are upregulated in the B cell population relative to all other cells, and those genes that are downregulated in the B cell population relative to all other cells. So I hope that's pretty clear, but again, for those that are new to this, it can get a little bit confusing, so please do feel free to post your questions. Gene sets. All right, so we've talked a little bit about gene sets. We saw one example there, but just to get into as much detail as possible here, these are going to be illustrated at the top of SeatGeek's workspace in the gene sets area above the groups panel. And gene sets can be exported. They can also be imported if you have gene sets that you've defined in another workspace, or if you have gene sets from the literature that you want to utilize in your analysis, you can add them to a list. And the whole purpose of that list is to combine gene sets that are genes that are in some way similar. They might be upregulated for a given population. They might simply be dividing up your different omics analyses, like I had mentioned a previous data set that we looked at where we have antibody parameters as well as RNA parameters. We might want different gene sets to distinguish the two. So they have a lot of different uses, but they basically combine different genes that were that make sense to categorize that way. There's also collections of gene sets. Lastly, these gene sets can be used as parameters themselves. So we do that by creating a statistic based on the gene set expression within a given cell in order to create histograms, just like you would a normal parameter in SeatGeek. But we call these synthetic parameters. So here we're just visualizing what a gene set looks like in the workspace. Here we have the down and up-regulated B cell gene sets that we identified in that previous example. We also see that you can look at the genes contained in a gene set simply by double-clicking on it. It'll open up the gene set inspector window that we see two examples of here. And of course, as I mentioned, you can gate on the gene sets themselves. In this case, we're gating on those on a gene set of up B cells and down B cells, and we're taking the positive up B cells, those that are expected to be expressed more in B cells than other cells, and we're taking a negative gate on the down B cells gene set, so those that are expressed lowly in B cells. And of course, what are we expecting to find? Our B cells. So how could we show that the cells, again, this is cell view, this graph on the left. How can we show that the gate we've drawn actually identifies the B cell population we had illustrated earlier? Well, we can just do an overlay of these red events onto the entire data set, looking at our antibody barcoded parameters. And sure enough, what we see is that our B cell CD19 positive events are being highlighted here, excluding all the rest of the cells, essentially, from that gate. So this is a good kind of gut check, a validation that by gating on gene sets, we are actually getting the results that we would expect or anticipate here. And that's sort of differential expression in a nutshell. Hopefully there's a little bit more understanding of what this process entails now. Keep in mind that you could do this comparison between any set of populations that you chose. It could be control versus test. It could be treatment versus untreated. It could be just different patients. If you have further questions, I would recommend strongly that you check out the documentation, docs.flojo.com/SeqGeek. We have plugins. I illustrated the Seurat, just a little bit, the output of the Seurat plugin. That's available at exchange.flojo.com. Plugins just add extra functionality and features to SeqGeek, so they can be really handy to have available. Tech support's available at SeqGeek@flojo.com. And we have 30-day free trials that can be extended for people that are interested in trying SeqGeek with their own data. And that's the end of my slideshow. I thought, kind of as a finale here, I would try to do a little bit of differential expression analysis live in SeqGeek so that people can see exactly how that's done. So here I've loaded some data directly from BaseSpace. You'll notice that if you are using Illumina's BaseSpace application, there's an add from BaseSpace option directly within SeqGeek. And clicking this will allow us to add data that has been shared with us on our BaseSpace account. In this case, we're looking at some PBMCs. I've gone ahead and done some quality control. Quality control is available through this button here. You can open up some different quality control parameters. That's probably a topic for another day. But after having grabbed those cells I'm interested in, I've just gone ahead and run Seurat clustering on those cells, and we can see what that looks like here. Essentially, we have created t-SNE-X, t-SNE-Y in order to do the dimensionality reduction that people often will want to do in single-cell RNA-seq, and then the Seurat pipeline itself has run a k-nearest neighbors clustering algorithm on top of that graph in order to get these different clusters. We see some really nice distinctions there. Now, maybe I'm interested in this population here, the red population. I already know that that's going to be my final population, cluster 6. I can tell that this is our Seurat clusters because it's listed as our tertiary parameter for color mapping here at the bottom of the graph window. If I wanted to do differential expression just on these 57 cells, I should be able to do so. But what do I want to compare with? In this case, I probably want to compare with the rest of my 1,500 cells in this particular data set. And again, in order to do that, I'll go to the Boolean options, which have not been added to this workspace. So if I click on the ribbon icon, I get a list of all possible ribbons, including the Boolean option here, which I'll drag into the workspace. Now I have a Boolean ribbon available. If I click on the Boolean drop-down list, I can choose Make a Not Gate, and we can see that our 1,500 cells outside of cluster 6 are now available as a population. Now, we already have a graph window open for quality cells. I can see because of this blue diamond to the left of it. And we can begin differential expression by setting up our initial comparison. But what do I need to do to begin that initial comparison? I need to get out of cell view. So I'm just very simply click on Gene View. Now we're looking at genes illustrated as dots for a couple of different populations. These are not the populations I'm interested in, however. So for the x-axis, I'll choose those cells that are not in cluster 6 versus cluster 6 itself. And we can see a slight shift in the gene expression here between comparing between these two populations. And that's all well and good, but I actually want to look at the fold change in p-values between these two populations. And so in order to set up that statistical test, I need to open the volcano plot, which is available through this differential expression button. And there we go. This illustrates those genes, very few though they are, that are downregulated in this comparison, and those that are, looks like a numerous amount that are upregulated in this comparison. So let's go ahead and just gate on those genes, excuse me, and we'll call these up cluster 6. I'm calling this cluster 6 because we haven't done anything like cell calling yet, where we could potentially try and determine what this cluster actually represents biologically. But for now, cluster 6 will do. Now, how else could I view this cluster 6 differentially expressed gene set? Well, it's available here in the workspace in the gene sets area. Looks like we've identified 140 different targets or features within that cluster that are upregulated. And I can look at the list. Sometimes you'll see categorical parameters in here, which we should probably ignore. You may also see clusters, or sorry, features that are not associated with the biology of this set. So we probably want to ignore those as well as much as possible. We could edit this gene set and remove them. But for now, I think what I want to do is switch back to cell view and illustrate how this gene set can be illustrated within that plot. Maybe we'll go back to the initial t-SNE mapping. So in order to do that, I'm going to open up the wings here so I can change the x and y axes to t-SNE x versus t-SNE y. Cool, so now this is a familiar clustering that we've seen before. If I now wanted to view this gene set, of course it can be viewed as a parameter within the cell view. It's available, if we look here, up cluster 6 is right there. And if I clicked apply, we would no longer be looking at t-SNE x, we would begin looking at the genes that are up in cluster 6 as a synthetic parameter. But again, I don't want to do that because I want to keep this map available. So one thing we can do is check this box to color axis. Notice we've already selected the Seurat clusters as a tertiary parameter, but I already know that this is cluster 6. So let's actually use our up cluster 6 gene set, and we can see that a few of the red events in that gene set are lighting up this cluster. Something else we could do if we wanted to make this a little bit clearer, we might adjust the scaling of our tertiary parameter. And in order to do that, I'm just going to open up, I'm going to duplicate this window and look at just a histogram of that parameter. And you can see that everything is sort of smashed against the axis currently. I could try using a different statistic to view that parameter. If I click apply operations, we should be able to see oh, I see. Here we go. There, now that's a little bit nicer. We're still identifying some cells in this cluster as well as cluster 6, but I think we're getting a clearer idea of what these cells are that are up in cluster 6. Again, I can also rescale this, and maybe a log scale would be better to look at. Somehow I think that this window needs refreshing, so I'll try. There we go. So you can see that the scaling of this synthetic parameter dramatically affects the way that this is colored in the color mapping within t-SNE. And that should give you just kind of a summary of different things that you can try while doing differential expression analysis and ways of exploring your clusters further in SeatGeek. I'll maybe wait just a couple more moments. If anybody has questions, please do post those to the Q&A board here. Otherwise, I encourage you to reach out to taylor@flojo.com and we can set up a one-on-one demo. We can start to analyze your data, or if you have any questions, just send those over. All right. Thank you so much for your time. Thanks for attending. Hope to see you at the next webinar. Bye now.